Let us take a look on the formation of how mitotic spindles are formed. So if you look at a typical chromosome, you can see that it is basically a combination of two sister chromatids together, which are held together at the centromere. So this is the centromere. But then there are certain button-like structures around the centromere known as the kinetochores. 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 So kinetochores are quite different from centromere because centromere essentially a bunch of repeated DNA molecules. So they're like there are nucleotides that are repeated continuously here. But kinetochores are different because kinetochores act as a primary place where there is an attachment of something known as the mitotic spindle. Now, mitotic spindles are spindles that actually pull apart the sister chromatids at the you know in the anaphase, and so there is the separation from the sister chromatids, and then there is the movement of the homologous sister chromatids near the ends of the cell. So it's important to know, in order to know more about mitotic spindles, it's important to know how exactly a centromere is a, a mitotic spindle is formed, like from where does it originate from. So if you look at the diagram right here, you can see that the origin of a mitotic spindle takes place in a centrosome. Now, a centrosome consists of, so this is centriole number 1 and this is centriole number 2, and they're associated at a 90-degree angle. As you can see, the angle right here is 90-degree, and sorry, it's like 90-degree, and then it is associated together with the help of a bond. So there's a bond, and they're held together at a 90-degree angle. At the level, like when the cell passes from the M phase to the G1 phase, it begins to divide. So even before the kinetic phase actually. So at the late anaphase and before cytokinesis, there is a formation of something known as a pericentriolar body. Now, if you look at the pericentriolar body, first, before the initiation of the formation of the pericentriolar body, it's important to separate these two centrioles, and the separation of the centriole is brought about by separase. After the separation, we have the formation of small bodies known as the pericentriolar bodies. So this small body right here is also known as the pericentriole. And pericentriole are associated in the same way in which there is an association of the previous association that we saw in a 90-degree angle to the parent centriole. So this is the parent centriole right here. This is also a parent, and the association here is at about 90 degrees. Now, moving forward in the S phase, you can see that lastly, after the formation of the pericentrioles, there is a division. There is a division of the entire cell into entire structure into two centrosomes. So centrosome 1, centrosome 1, and centrosome 2. And each centrosome will go and incorporate itself into the daughter cell. All right. So, in order to let us learn how is there a formation of mitotic spindles. Now, before we do that, it's important to know that the formation of mitotic spindles happen with the formation of an astral-like projection that takes place from the centrosome. So, the structure that you can see right here is known as the centriole. The centriole. This is the centriole, and the centriole is usually found in the centrosome. And around the centriole, there is a mass. There is a small mass, the cloudy mass right here, which is also known as the pericentriolar matrix, or the PCM. Now, if you look how there is a projection of mitotic spindles outside. Mitotic spindles are nothing but microtubules, so they project out of the pericentriolar matrix in this form, and usually their minus n is associated with the matrix, and the plus n is the one that is actually projecting outside. Now, why is that so? The formation of microtubules usually takes place by the addition of subunits on the plus side. And this addition is mediated by something known as polo-like kinases. So polo-like kinases assist in the nucleation of the microtubules that originate from the pericentriolar matrix. Now, nucleation is a really slow step. So it starts off slowly and then there's a rapid elongation. Now, what happens after the elongation? After the elongation, there is an attachment, and there's an attachment, as you can see, of the mitotic spindle of the aster-like projections on the kinetochores. So these are the kinetochores of the chromosome. After the attachment of the chromosome, the next step is the movement of the chromosome. If you look at the cell, there's an equator, and then here, for example, here is the centrosome, which consists of the centriole, and there's a projection. For example, the chromosome is situated here, and then there's a projection of the mitotic spindles right here. The next step is to move the entire structure near the equator, near the poles, and there is there's a pulling of this entire structure towards the centrosome, which migrates first here and then pulls the entire structure towards the pole. And this is mediated by something known as a motor protein. So these small structures, as you can see here, they travel across the microtubule, and then they pull this entire structure towards the centriole. So these are motor proteins, mostly consisting of kinesins.